Hey guys, sorry, I got the headset on again because when I listened to part of that last lecture, the sound quality kind of was crappy. So I'm hoping this will help a little bit. Okay, we're doing immune system 1. And go. All right. Okay, let's go ahead and finish this pre-lecture assignment. This is immunity. Immunity is the capability of killing or removing foreign substances, pathogens, and cancer cells from the body. I'm going to switch that to black. All right, so we're going to cover innate immunity a little bit in the lecture, but here's some background information on it anyway. Okay, so innate or non-specific immunity does not require previous exposure to initiate defenses. I'll go into this in a little bit more detail in just a minute. Innate defenses occur immediately or very shortly after infection occurs. With innate immunity, there is no memory or recognition. That an intruder has attacked before, and therefore no immunological memory of the attacker. Okay. The body has built-in barriers, physical and chemical, that serve as the first line of defense against an infection. The skin or the intact skin is generally an effective physical barrier. Notice it says intact because, as you know, if we have a cut or some kind of injury, bacteria can invade. Physical barrier that prevents infection. The skin also has chemical barriers in the form of sebaceous glands. That's what oil glands in the skin are called. That weaken or kill certain bacteria on the skin. Mucous membranes lining the respiratory, digestive, reproductive, and urinary tracts are also considered physical barriers. To immunity. A significant chemical barrier to infection is created by the normal flora, the microbes, usually talking about different types of bacteria that are present in various parts of the body, reproductive tract, mouth, large intestine. Okay. All right. So let's talk a little bit about innate immunity. I'm going to fill out the notes kind of like I did last time, and then I will go over the steps in a little bit more detail. Okay, move you over there. So remember that the skin, physical and chemical barriers are considered the first line of defense. The second line of defense then is going to be innate or non-specific immunity. Remember that this takes place, or this engages pretty much immediately upon exposure to a pathogen, okay? What happens is, let's say if you cut yourself on something, and that's something that you cut yourself on has bacteria on the surface, okay? So not only do you have a cut, you have bacteria that's invading your now broken skin. When that happens, something called histamine gets released from the damaged tissues, okay? Or from another type of cell in the blood called mast cells. Histamine produces an inflammatory response, okay? Or an inflammatory response is usually where you have kind of a congregation of blood cells, and different components in blood that all rush to the site of something. It causes swelling. So increases blood flow, which causes this swelling, pain, heat, can feel hot at the site of the wound, okay? And then we have another reaction, which is actually about the capillaries, which if you remember are the tiny, tiny blood vessels. So it makes the capillaries more permeable to white blood cells. In other words, it lets the white blood cells flow in and out of the capillaries with more ease, which is needed because those white blood cells are using the capillaries and the blood vessels as subway system to reach the site of the injury, okay? All right, so again, histamine released from damaged cells or damaged tissues or from mast cells. The histamine causes this increased blood flow. It makes the capillaries more permeable to white blood cells. And then this increased blood flow that results brings in our macrophages, which if you remember are big eaters. They engulf foreign particles. Okay. Then we have neutrophils and monocytes. Again, monocytes mature into macrophages. We have them arrive at the site as well. And they are going to also do their part. So they're going to engulf some of those invaders as well. Hang on a second. I'm going to cover this because that's not what I wanted to put there. No, that's not what I wanted to put here. I wanted to put here cytokines. These are like chemical signals. Okay. Work. I got a little ahead of myself. I apologize. Let's try that one more time. All right, so they're going to release these chemical signals that you can kind of think about as being chemical flares, right? They basically tell other white blood cells in the area, hey, the trouble is over here, come quickly. Okay, it guides them to the location of the injury. All right, now I can get to this. Again, sites. And then all right. So the neutrophils and monocytes neutrophils can assist macrophages in engulfing the invaders, but then they can also release cytokines as needed. So if more backup is still required, even with this assistance, then they can release cytokines, those chemical flares, to bring in more. All right, other types of parts of the innate immune system. We have what I think are really cool, complement proteins. Complement proteins have the job of finding bacteria, poking holes in it, and destroying it. Because when you poke holes in bacteria, all of the fluids that are inside gush out and the bacteria dies. complement is pretty awesome. Another group of proteins that participate in the innate immune response are interferons. Okay, how this works. So first off, remember that viruses are not alive, okay? They have to hijack the host's cells and the organelles in those cells in order to make more virus, okay? So if a virus invades and it gets into your cells, it's going to take over all of the organelles in those cells to make more viral proteins instead of making the normal proteins that your body would make. Okay. When that happens, when a cell is invaded, it recognizes that it's infected and it releases a protein signal, not cytokines. It's a different type of protein signal. And it says, hey, I've been invaded. That's where the interferons come in. They come in and they bind to the receptors on the uninfected cells that are all around the one that sent out the signal, okay? When they bind to those cells, the interferons basically are telling the cells, hey, your neighbor just got invaded and you need to ramp up your defenses, okay? I think complement proteins are cooler, but maybe that's just me. All right, let's talk about adaptive immunity. Adaptive immunity is specific immunity. Remember I told you, and also from your pre-lecture assignment, it talked about the fact that your that innate immunity and the cells, the white blood cells that are involved in it, they don't remember a past infection, okay? So you can have an infection, get over it, 4 weeks later, come down with the exact same infection, and your innate immune system isn't going to remember it, right? The innate immune system is there to simply destroy anything and everything that doesn't belong. So it's not going to come in contact with some virus that you just had and be like, yeah, I remember you. You were just here a couple weeks ago, right? Yeah, no clue. They're just killing machines. They're out there to destroy anything and everything that doesn't belong. Because of that, they can respond immediately because they're not looking for anything specific. Well, with adaptive immunity, the cells involved in adaptive immunity do remember. They do remember past infections, okay? That means that it takes them a little longer to respond. You'd think it wouldn't because they remember, but it does because they might remember, but their army isn't ready to attack just yet, okay? Because it takes about 7 to 10 days to get that immune response up to full strength, okay? So if a pathogen gets past the first line of defense, the physical and chemical barriers, past innate immunity, then it's going to be dealt with by the adaptive immune system, okay? All right. All right, so again, why this takes a minute. They have to recognize and identify the pathogen, and then they have to build up their response. There, where are the same white blood cells that are needed, okay, to kill them. So that's why it takes time, 7 to 10 days, like I said. Okay, so what types of pathogens would they recognize? Pretty much all kinds. You've got bacteria, viruses, you could have parasites, some cancers. Cancers, as we learned in the last chapter, are harder because the body still recognizes them as self. So unless the cancer cell is sending out a signal that it's been compromised, it may go unrecognized by the body. Okay, remember that when it comes to our cells in our body, we have markers on the surface that sets them apart as belonging to us, right? So the adaptive immune system recognizes those, the ones with our name tag on it. Okay, all right. So we talked about some of the cells that were involved in innate immunity, macrophages, monocytes. But now we're going to talk about the ones that are part of adaptive immunity. And those are lymphocytes. And lymphocytes consist of the B cells. Okay, we're going to talk about the B cells first. Notice here that it says that B cell lymphocytes are part of antibody-mediated immunity. What this means is that you have B cells that are left over from past infections. So after an infection has been cleared up, some of the B cells stay behind because they're going to be looking, as the assassins that they are, they're going to be looking for any sign of that invader again. So they circulate in the blood. When a memory B cell finds the invader that it recognizes, it chemically binds to it, and then that B cell replicates and makes copies of itself. That process of making copies is called amplification. Okay, so that B cell is going to amplify itself, making a bunch more of it that can find the same antigen and destroy it. Again, that's part of the reason that there's a delay in the response, because B cell finds it, recognizes it, identifies it, but then it has to build up its army to go and find all the other antigens that are in the blood to kill. Okay. All right, so clonal expansion, this leads to our amplification process once an antigen is recognized. So basically, spell out the word B. The B cell found the antigen amplifies. Okay, makes more of itself. All right, so once that amplification process has taken place, the B cells are going to mature into either plasma cells either plasma cells or into memory B cells. Okay, plasma cells, their job is to secrete antibodies that match the antigen. Okay, so obviously, if you think about an antigen and an antibody as kind of being like puzzle pieces that have to fit together, the plasma cells are responsible for creating antibodies that have the correct structure and composition to attach to the antigens. Okay, because of this, we go back to our organelle cell lecture from beginning of the semester. Plasma cells have lots of rough type because these are proteins, right? And ribosomes build proteins, and if we've got ribosomes embedded in the ER, you're going to be assembling things that have a lot of protein in them. Okay, our memory B cells, again, these are the guys that are going to stick around and remember past infections. Move this up a little bit. Maybe I won't. Right there, okay? If an antigen is encountered again by a B cell or a T cell, but if it's encountered again in the future, the body is able to respond faster and harder, okay? So rather than having it take the 7 to 10 days it did upon first infection, it can cut that down significantly. 3 days, 5 days, something like that. And so not only does the body respond to it faster, it usually is able to destroy it faster, so you're not as sick for as long. Okay? All right. Okay. So we talked a little bit about antibodies in the white blood cell chapter. The antibodies, again, are this Y shape. They have a binding site that matches the structure and chemical composition of the antigen so that it can bind to the antigen. This is the enemy's head on a pole type thing, right? Once an antigen has been attached to an antibody, we've got 4 different things that the antibody can then do, okay? First, they can call in my personal favorite, the complement proteins. Sometimes just called complement. They can trigger phagocytosis. They can prevent the function of the antigen and of the pathogen because, you know, it's chemically bound to the antibody, meaning that it can't function because the antibody is holding on to it. And finally, it can also inhibit the release of toxins from the pathogen. Okay. All right. So how are antibodies used in medicine? Well, we have monoclonal antibodies that can be used in treatment. Antibodies that originate from a single B cell. That's what we're looking at with monoclonal antibodies. So remember that I told you on the last page that when a B cell recognizes a pathogen, it makes more copies, it amplifies itself. That's what we're looking at here. So what happens with this? We've got our antibodies that are what happens with this then is if a person becomes ill with something for the first time, we have our 7 to 10 day response before the innate immunity, before the adaptive immune system can get up and functioning. And during that time, it's possible that the infection can overwhelm the person, right? They can die before the immune system can really get going, or they can become very sick. You can actually give a person antibodies and have those antibodies immediately begin to search for and destroy the infection, okay? So like with COVID-19, you can take the antibodies that a person that's already recovered from COVID-19, you know, they've already recovered from it, you can take their blood, take the antibodies, and actually use those then to inject into another person. And so you immediately ramp up the number of antibodies that can fight that infection. So while your body is still trying to make more of those, it's still in the amplification process, you just took a bunch and put it in the body that's ready to use, okay? Your troops are still trying to get together, build up their strength so that they can fight. You just brought in an influx of troops from another source that are ready to go right now, okay? These could also be used in diagnostic tests or viral testing. One of them is to detect HCG, which is a pregnancy hormone. It's human chorionic gonadotropin is what it stands for. And then of course this can be used in HIV testing as well. Some cancer treatments, this is kind of based on that immunotherapy topic from the last lecture. You can have specialized cells that recognize cancer and be able to destroy those. I don't mean specialized cells, I mean specialized antibodies. Okay, COVID-19 treatment, like I said, back up towards the top, monoclonal antibodies. Okay, let's talk about T cells. If I have to choose between T and B cells, T cells are awesome. B cells are great too, don't get me wrong, but T cells are my favorite. If they were a house in Harry Potter, T cells would be Gryffindor, and B cells would be Hufflepuff. Still great, but you know, can't compare. All right, so T cell lymphocytes, unlike B cells, T cells are part of cell-mediated immunity. What that means is they are circulating in the blood, they can remember past infections, and they can destroy invaders. But they can't bind to that invader first. A B cell can be out circulating in the blood, find an invader, bind to it, destroy it, and then start the amplification process. A T cell has to have an invader presented to it in order for its process to start. Okay, kind of like the king saying, bring me his head on a silver platter, right? Well, that's what the T cells are demanding. And the lucky white blood cell that gets to bring the metaphorical head are macrophages. So macrophages, part of our innate immunity, wandering around, find a foreign invader, they don't recognize it, but they kill it because it shouldn't be there. When they kill it, they present the antigen to the T cell. Hey, my lord, I have something for you. Here's its head, right? The T cell is then going to take it, binds to that antigen, and then the T cell expansion process takes place. I'm going to be specific here because we're going to get into 3 different types of T cells. In this case, it's the helper T cell that's going to bind. Okay. All right. So the macrophage finds an invader, destroys that invader, takes the parts of that invader to a helper T cell and says, look what I have found. And the helper T cell takes that antigen and then starts the clonal expansion process. And it does that by releasing cytokines, which, remember, it's a chemical signal, okay? When the cytokines are released and we have clonal expansion begun, we can have 3 different types of T cells produced. 1 helper Ts, which personally I refer to as the sidekick T cell, because the most awesome T cell is the cytotoxic or the killer T cell. Okay. They're the real awesome one. They're the superhero here, right? The helper Ts, it's the sidekick. What do? I'm going to release cytokines, which is super important. Not as awesome as the killer T that's going to actually go in and destroy cells that are already infected. The third type is our memory T cells. Do the same thing as memory B cells. They remember past infections and are ready to get the process rolling after an antigen has been presented to a helper T. Okay, fun. Last little thing we have, which I actually really like, find it helpful. This diagram thingy is going to help us see what causes what. Get that out of the way. When it comes to our T and B cell production and the T and B cell immune response. So this is part of the adaptive immune system here. All right, so we have a macrophage. Okay. And again, remember that while a macrophage is part of the innate immune system, when it's presenting an invader to start the expansion process for T cells, it's actually assisting the adaptive immune system. So they work together. All right, macrophage eats an invader and presents the antigen to our helper T cell. Okay. When that happens, the helper T cell, or the sidekick T cell, is going to do what sidekick T cells do, and it's going to secrete cytokines. Okay. Right now I'm going to focus on the right half, which is the T cell response. I'll do the B cell in a minute. Okay, so for T cells, once the cytokines have been secreted, it's going to activate our T cell clonal expansion. Okay, we know that when clonal expansion starts, we have three possibilities. We can either get more helper T cells, sidekick T cells. We can get the truly awesome, the truly amazing cytotoxic killer T cells. Okay, or we can get our memory T cells. Okay, all right. Helper T cells, hey look, it's going to start the process over again because it's going to secrete cytokines because sidekick T's secrete cytokines. There you go. And our cytotoxic T cells are going to kill cells that are already infected. Goodness. Okay. Ta-da! That's on the T side. Let's go to the other side and look at our B cells. Remember that B cells do not need a macrophage to present an antigen to it. So you'll notice that the arrow for a macrophage eats an invader does not go to the B cells, it only goes to the T's because the B cell can find it, identify it, kill it by itself. When a B cell binds to an antigen, we get our B cell clonal expansion. Okay. We know that when clonal expansion happens for B cells, we have two options. We can either have plasma cells, which produce antibodies, okay? Or we have our memory B cells. And like memory Ts, those are going to stay in the blood in small numbers and circulate. I should point out that following an infection, a lot of the memory cells, B's and T's, are broken down, okay? Because you don't keep all of those cells circulating in the blood if there's no reason. You have enough that there's a constant watch happening, but you don't have huge numbers, okay? All right, you guys, that is the end of Immune 1, and I will see you soon.